Good morning, everybody. Okay, here we go. I have a short video to start with, and we'll move on from there. Good morning, everybody. Here we are, getting started on this new normal of ours. And so this is going to be our first lecture since we started this remote thing. So I'm grateful for you to give patience and grace to me as I navigate through this. I have had lots of trouble trying to get the Zoom and the recorded PowerPoint to actually play videos. So you know how much I count on videos in these courses, and we really, enjoy the video section of this. And so this is a way I have figured out to do it, so you can see these videos, and that is actually to put them through YouTube. Yeah, so here I have a YouTube channel. Yay, YouTube star. Here we go. All right, so you're my guinea pig class. We're going to see if this works. I'm going to send this to you, and I would appreciate it if you would let me know how it went. Okay, so this is the class that is going to be for our first Tuesday back, and I'm going to go through that on our PowerPoint in just a minute. So this will be a PowerPoint presentation, just like we always do. It's going to be through YouTube. You will be able to pause it whenever you like. So sometimes I go too fast, and so you can pause it and write down the notes. So you'll have your lab book in front of you, ready to take the notes just like we normally do. And at the end, we'll have a little lab that we'll do that was kind of a short lab for this setup anyway. Okay. I do need also feedback. How's the sound? Can you hear me okay? Is there enough light? I put a bright light right over here, so hopefully that's going to keep us where you can see me. And then you can hear me while the PowerPoint is going. I have had a little reverberation going on, so I'm going to experiment a little bit with that, and that's what y'all are here for me today, so I can experiment with you. So you know that we have had our trip to the coast canceled, and I'm so sorry about that. I had it all set up, and the University of Texas contacted me and said that they are canceling all spring tours, all of them. And they're considering gearing back up in the summer, but they don't even know that right now. This is just unprecedented, what's going on with us, and it's just crazy. Yeah, so we all have to get through it together, and I don't know how we can make up that field trip. It was sort of a special time for us to do. But like I told you in my email, I do intend to cook for you still, so we'll have to figure something out. I am absolutely confident we will end up getting back together again in classroom. I just can't imagine. I'm just such an optimist. I can't imagine it would be any other way. Okay, so tune in now. You're going to go ahead and just watch this. I'm going to send you a YouTube link. You should already have that. You will click on that, and if you want to make it full screen, it'll be just like you're watching it from the screen there. Okay? All right, y'all. See you in a minute. Okay, so here is our new schedule. As you can see right here, we had second spring break, right? And then we're going to jump right back in. We finished 13A the last time we were together, and we'll be on 13B this time, still learning about the epipelagic. We'll get reacquainted with that here in this lab just in a few minutes. We're going to move on to the deep oceans, and then we're going to be looking at human responsibility with the oceans right here in these last two modules. If this tasting party right here needs to be moved, if we get back together like for the last 2 weeks or something, I'll move that. Okay, but I do expect y'all still to have a project to do. The project can be anything about marine biology. It just needs to be, it could be put into a PowerPoint like this, and you could, if we don't get back together, you could be sending that to me and I could look at it that way. We could do a Zoom that week and have everybody, once you've sent them to me, I could then put them in a big compilation, I believe, and we could watch them all together and y'all could talk about them. We could do something like that. If you don't want to do a PowerPoint, then we can do the threefold board like you would be doing at the science fair or something like that. That'll be just fine. It just needs to be about marine biology. You can choose one of the cool animals we've learned about, one of the newer processes of traveling through the ocean for research, like I had a student one time do one on the submarines that would go down and do some research, something like that. Also, you'll see our final exam is scheduled here. I do plan on having a final exam. Sorry to tell you that, but yes, that's the deal. And if we have to send that home, then we'll figure out a way to do that. All right, so here we are, the epipelagic zone. It is 13B, Lab 27, page 88 on your lab book. So you should have your lab book out and open to page 88. And let's see what we remember about the epipelagic zone. All right, so what is the epipelagic? You can see right here, it's this area at the top, right? So it goes down to about 200 meters. It's the area where light can easily get to. It's the photic zone. That means light gets there. And we have lots of life teeming in this area right here. Remember, we don't think of it as any kind of bottom to it. So all the animals are floating or swimming here. We have zones beneath it, which we will cover in module, I believe, 14. Right here, we see the mesopelagic, bathypelagic, abyssipelagic, and the hadopelagic. And you can see how we're going to learn about critters down here get very, very strange. So, epipelagic, 200 meters. That's probably a good quiz question right there. How far down does the epipelagic extend? You want to remember that. Here are some of the creatures we find in the epipelagic. There at the top, the swimmers that are up there. They will sometimes go down a little bit deeper, but for the most part, they're considered the swimmers, the nectin, the epipelagic nectin. And as we get down deeper, you can see some of those pictures of those strange creatures there. So here's our first blank we're filling in, the epipelagic nectin. If you remember, nectin means they're swimmers. Okay, so we have some strong swimmers who use speed and numbers to ensure their survival. This picture you see to the right is a giant school of anchovies. And they hunt in huge packs. They eat phytoplankton and zooplankton. So that is their food source, but then they in turn become the food source for other larger fish. And you will not believe the massive schools these guys travel around in. I mean, they're intense. Another fish that was similar to this would be, of course, the sardine. They're a little smaller, and they also swim in these massive, massive schools, hunting phytoplankton and zooplankton. So the nectin, remember, are the swimmers. What do they eat? Most of them are carnivorous. They're predators, and they go after other fish. There are such a thing as planktivorous nectin, and they eat zooplankton, and many of those are strong swimmers, and some of them are actually quite large. So we're looking at two planktivorous groups that are opposite in size. The small groups, which would be our sardines and our anchovies, and then our giant ones, our baleen whales, our whale sharks, our basking sharks. You're going to see those. Those we'll get to. You see how they are massive fish that survive off of plankton. So planktivores, let's start with those. Here are the anchovies, right? They are swimming around. And a giant school right there. We can see some fish over to the left who probably want to make a meal of these guys. This is a giant school of sardines. There is a diver in the middle there, swimming right through the middle of the school. I'm going to bring it in a little closer for you to see those fish. See them in there? Crazy. There's so many of them all compacted in that thing. So the idea here is they're getting protection in numbers. So the ones that are in the middle there, so that's kind of a solid clump there. The ones in the middle are being protected by the ones outside of them. And so the ones on the edges, those are the most vulnerable guys. We're going to see in just a minute how they become quite vulnerable. Here's another picture of some sardines. This picture is kind of fun. We have some surfers who are going out. Sometimes these schools can get very close to where swimmers are, and they'll actually be swimming. The fish will be swimming all around the swimmers, and it's kind of creepy. But if you start to move toward them as a group in mass, they will all move away. So that's what's happening here. The surfers are swimming out, and as they're splashing, they are pushing those fish away. Those fish are swimming away from them, and you can see it is such a massive change because where the fish aren't is the nice clear water, and where the fish are, it's absolutely dark there. Here's some of our larger marine nectin. They're rapid swimmers. We see dolphins, sharks, fish, even some squid and things down there. They mostly have very hydrodynamic bodies, streamlined bodies to get through the water quickly. The whales are big, the whale sharks, and some of those end up being planktivores, and I'm going to show you a video now of the feeding frenzy. So when we have some of those large, giant schools of smaller fish, like the sardines or the anchovies, you're going to see that it will attract predators, and they will come to find food.
Even when more sea lions arrive, they can't seem to break down the sardines' coordinated defences. With a shoal this big, the sea lions need to isolate a smaller, more manageable group of fish. But with so few predators, the fish still have the advantage. All the sea lions can do is keep the sardines at the surface and wait for others to join them. Tuna. Their arrival changes everything. Tuna attack from below, cutting off the sardines' escape route down to deeper water. Next to appear shearwaters. Excellent flyers, but also surprisingly agile underwater. With so many predators attacking from all sides, the advantage starts to shift away from the sardines. As the fish pack ever tighter, their shoaling strategy now makes it easier for the hunters. Copper sharks. They've scented blood in the water. Surprisingly, perhaps, the predators never attack one another. They work together to corral the ball of fish, taking turns to grab a mouthful. Common dolphins. As the shoal gets ever smaller, each sardine scrambles desperately to hide in the middle. But now there's no escape. A brooder's whale finishes off the feast. Tons of sardines devoured in less than an hour.
Okay, so there we go. So back to our planktivores, guys. Here's a basking shark. So remember, the planktivores are the creatures who will consume plankton. All right, so some of those would be small, but some of them are quite large, like this basking shark. All right, this is an interesting creature. They call it basking because it stays somewhat near the surface and 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 moves very slowly through the water, almost like they're basking in the sun. And the whole time their mouth is open. And as their mouth's open, the plankton and stuff, he'll just collect up and vacuum up whatever's in front of him. He's got all sorts of um, gill rakers in here. You can kind of see them right there, and those will capture those into the gills, and then he can squeeze the water out of that and ingest the creatures there. The whale shark is also planktivorous. Notice its mouth is also open. So it's going to be open collecting all the plankton that it comes across here as it swims through the water. Now, these guys are some of the most fascinating, the baleen whales. First of all, they're some of the largest creatures on the planet. The blue whale is the largest animal on the planet. And it is bizarre to think that that creature stays alive by eating nothing but plankton. All right, so they have this fancy type of structure in their mouths called a baleen. So they don't have teeth like a regular whale does, like you think of an orca or something like that. That's a toothed whale. These are baleen whales. We're actually going to talk more about whales at the end of this lecture just because I would like to bring those into our discussion. But these guys right here all have this fancy mouth part called the baleen. I'm going to show you a picture of that now. So you see that crazy looking feature there. It's sort of like a brush that hangs down, like almost like a giant toothbrush that's inside their mouth. And what they will do is they will gulp water into their mouth, and then they will squeeze their tongue up to the top of their mouth and squeeze all the water out so that they trap inside the baleen, inside those little bristles, they trap the plankton in there. Here's a closer up of the baleen. You can see it there with the hairs on the end. It's sort of like hair material, keratin, like our hair is made of right there. And you can see how things are getting stuck inside those little crevices there. All right. Another strategy for the creatures in the epipelagic is staying in the epipelagic, okay? Because if they sink too far down, they'll get into the dark regions. They'll get to a place where they don't have a lot of food. And so they need to stay afloat. So we have seen already some strategies for this. If you remember the lab we did with the clay, we had a clay ball and we dropped it, and then we had the clay ball with the toothpick sticking out of it, and it didn't fall as fast. And so having the projections on them is one way to stay afloat, to give them some buoyancy. There are some more ways as well. So we can see here, we have this sea sparkle right here, that they have these vacuoles inside of them, and it gives them buoyancy. Sometimes those vacuoles can be gases from inside them. Sometimes they could be air bubbles, basically, that will cause them to float. Right down here, this violet snail is kind of crazy. It shoots bubbles up, and these bubbles have sort of a mucousy substance in them, what causes them to float, and the snail just hangs on. He just holds on and floats near the surface. We'll see why in a few minutes. The glass squid has a little chamber in here filled with ammonia, and that ammonia, you can manipulate how much is being released, and they can cause the pressure to change inside their body, giving them more buoyancy. We'll see that in our lab in just a few minutes. Right here, we see those projections, like our clay pieces that I talked about earlier, that will help them stay afloat. And then down here, our fish, of course, have a swim bladder that gives them buoyancy. They can manipulate that. Some fish, of course, don't have it. The catfish, for instance, doesn't have one, and so they tend to sink. And of course, sharks. We looked at our shark, and there was no swim bladder. And so we know that for our shark to stay aloft, to stay afloat, has to keep on swimming. Here are some other strategies right here. We can see with our nautilus up here. Remember, it's called the chambered nautilus because there's these chambers in here. And we saw one. We looked at it. Remember, it had those little holes to communicate between the chambers, and they can pump gases, air, if you will, into several of those chambers and make them buoyant, and they'll float. They can pump it out, and they will start to sink. So that gives them sort of some control over where they are. We have these squid-like creatures here, this one with the pin, like our squid that we dissected, and they can use that pin for buoyancy right there by just manipulating body fluids inside there. This guy actually has a little shell type structure with some chambers that, like the Nautilus, can have change due to the gases that are put in there. And then, of course, we have our swim bladder in our fish. You can see right here they can regulate how much gas is in the swim bladder through their circulatory system right there. And then we'll see how they can float or sink, depending on where they want to be. There is a lab in your book. It's experiment 13.2. It's very, very similar to the clay project we did earlier. So you can see here, you're going to drop a piece of clay in, one round and one flat. You'll see the difference there of how they fall to the bottom. Then you'll have a round one and then one with toothpicks and this kind of thing. You can go through those trials. And if you take a picture of a couple things and send that to me, I'll give you extra credit for doing this little lab right here. So you see at the bottom, try to build an organism that completely resists drag and floats. Okay, so you might want to think of maybe getting some air bubble in there or something like that. It might be a fun little experiment to do. All right, so here we can see some more pictures of buoyancy. We have the fat and the oils right here in this critter, a little gland right there, and they'll have oil floats on water, and so this will cause them to float. There's also these projections right here that kind of give it a little bit of floating ability as well. And our other creature over here has apparently some swim bladder, some gases in here, and some projections that allow it to stay up there. We learned about the by the wind sailor. It has its own strategy for staying on top. In fact, it stays on top of the water completely, right? And has that sail that causes them to blow about in the wind, and they can move across the surface. Now, it turns out our violet shell over here we saw earlier with its mucus bubbles, guess what? Guess what it's hunting? It's hunting this guy right here. So where does it need to be? Well, it needs to be on the surface. And when it does come across one of these by the wind sailors, it'll have a tasty snack. So our predator-prey relationships in the epipelagic are interesting. They have to hide and seek and stay alive. Okay, so remember we're looking at no bottom. We'll be looking at a little bit of camouflage here, but for instance, our first slide, this is called the crocodile fish. So even though this picture shows him on the bottom, we don't really think of him as a bottom dweller. If he's swimming and there's a fish above him and looks down, he will blend in with what's below him. So if he's near the bottom, it'll blend in that way. And so that'll give him some good camo right there. So that's some pretty good camouflage. Now he can be camo as a predator, lying in wait, or his camouflage is to protect him from becoming prey. Here's our glass squid again. We know that it's got that little gland in there that allows it to float. But it also, from underneath, if you're underneath it looking up, it'll blend in with the brightness of the sky. So it'll be more difficult to see. It's almost transparent, giving it, you could call that a type of camo. So other ways to avoid getting eaten. All right, number one would be speed. If you're fast enough and you can get away from your predator, then you're going to survive. So a lot of these fish out there in the big ocean have dark muscles with large amounts of myoglobin in their blood, which gives them dark meat. So if you think about like a salmon, how that meat is very, very dark. This enables them to move quickly even in the cold waters. Counter shading is probably the most important in the marine world because almost every creature does exhibit this characteristic of counter shading. Fish, most fish, our shark had this. I'll show you a picture of that here in just a minute. And what it basically means is on the top side, they're dark so that if there is a predator above them looking down, they will blend in with the color of the ocean. Whereas on their belly side, on their ventral side, it's light and silvery. So if there's a predator beneath them looking up, it will actually blend in with the brightness of the sky. All right, so counter shading is incredibly important. Another strategy would be vertical migration, and that is to change where they are at any given time. So these creatures will move up and down the water column to avoid predators. We'll see some slides here in a minute, how in the nighttime they'll move up, and then in the daytime, they'll actually move down to where it's darker so they can't be seen as easily. So here's a fast fish. This is, this is called a sailfish. And it turns out, I guess the sailfish is the fastest fish in the ocean. It goes about 68 miles per hour. So that's fast. So they can get away from most predators. And of course, as a predator, they're very successful in catching their prey. Here's a great example of counter shading. You see our shark here, and the dorsal side here is dark, so if we are above it, and it'll look very similar to the ocean that it swims in. And then underneath, it's very light and silvery. So from underneath, they blend in with the sky. Our vertical migration, I think this is what you're going to write down in your book, that the zooplankton will migrate up to the surface at night to feed, and then they will descend into the deep water to avoid predators. So that's your official definition that you're going to write down. If you want to pause this slide so that you can take time to write all those words, I know y'all like to write all the words, you can do that. But I'm going to show you a few more slides of this because that picture is not great. This shows almost like a scale of the type of life. So we see those dots right there represent that at nighttime they've moved up here, and they're fewer down here. But at twilight, they all move down to the lower depths to avoid the creatures up there in the daytime. And here's another picture of it. So at nighttime, here they are up there feeding on, guess what? Those phytoplankton that are floating there, right? But in the daytime, they'll go way down here and wait. They'll just wait until the evening to come feed on these guys who are going to proliferate during the daylight hours. Notice they do go out of the photic zone and down into the mesopelagic area just to hide. We're going to watch it's a fun little video on this.
How did I get here? Well, it's a stranger story than you might think. I came from a world of drifters, a place few humans have ever seen. The world of plankton. I came from a batch of 1 million eggs, and only a few of us survived. When I became a larva, I moved among other drifters. My fellow plankton came in all sizes, from tiny algae and bacteria to animals longer than a blue whale. I shared my nursery with other embryos and juveniles, from clams and crabs to sea urchins and anemones. We drifting animals are called zooplankton. The most common animals here are copepods and krill. You could search the world over, but you'd never find a place more diverse than my childhood home. A teaspoon of seawater can contain more than 1 million living creatures. It can be a pretty tough existence, though. Trillions are born here, but only a few make it to adulthood. He may be no larger than a pinhead, but this crab larva is an arrowworm's worst nightmare. Epic battles between carnivores like these are just one way to get food. But the real powers of this place come from phytoplankton. Single-celled life that transforms sunlight and carbon dioxide into edible gold. phytoplankton are the base for the largest food web in the world. During the night, many animals like me would rise up from the depths to feed on this sun-powered feast. I was part of the largest daily migration of life on Earth. During the day, I'd return to the darkness where I'd join my bizarre companions. Cannibals, like this sea butterfly mollusk that eats its next of kin. And comb jellies that beat cilia like rainbowed eyelashes. Some of these snare their prey with sticky tentacles, while others just take a bite out of their cousins. And siphonophores that catch prey with toxic fishing lures. But my favorite would have to be the crustacean Phronema. Its monstrous looks inspired the movie Aliens. It can catch tiny bits in its bristles, but prefers larger prey like salps. With 2 sets of eyes, this female prowls the deeper water. Prey in hand, she performs one of the strangest behaviors in the entire animal kingdom. With body parts from her victims, she delicately assembles a barrel-like home, feeding her young until they can drift off and survive on their own. Best of all, they make the perfect snack for a small fish like me. Here among the plankton, the food web is so tangled and complex, even scientists don't know who eats whom. But I do. At least now you know a bit of my story. There's so much more to me than just a tasty meal.
So we see that I was talking about in that video about the food web and how that web is very complex. So remember, when we're talking about food webs, food chains, things like this, we're looking at the flow of energy. It requires a whole lot at the bottom of a food pyramid to support the life from the apex predator there at the top. Okay, so we're always looking at that flow of energy. So of course it begins with the phytoplankton. They are performing photosynthesis. Then we move on to dissolved organic matter, the DOM, and the smallest zooplankton, the bacteria. These make energy available to the rest of the food web. So our dissolved organic material, and dead organic material as well, diffuses out of phytoplankton and waste products from protozoans. So that's kind of the stuff that ends up, and then furthermore, some of the dying creatures as well. It's eaten by bacteria, who then in turn get eaten by the protozoans, who then get eaten by the zooplankton. And then we can see how that would put the energy into the food web, because then they will be consumed by some of the planktivorous creatures we've spoken of. Then those creatures are then eaten by larger creatures. So we see this loop. So we see how the microbial loop, though, is kind of in the middle there. That's going to kind of sustain this whole deal. So right here you can see we have bacteria in the ocean. We also have the broken down parts. So our organic material, here it's called the POC. It's got the carbon in there as well. It's sinking down. The bacteria will use it, and then they will go through respiration, and we have this whole cycle. So you're going to need to draw something in your book, but don't draw this one. I think this one's a little too complex. We'll draw a different one right here. But I wanted you to see how our bacteria are down here, and then our zooplankton are above, and then our phytoplankton are up there, definitely up in the photic zone, right up in there. And so we can see also vertical migration taking place between those, which allows the zooplankton to be near that bacteria and see that loop. We're going to see a video about the loop, and then I'll show you what to draw. So what is it? The flow of energy in the epipelagic, beginning with the phytoplankton and dissolved organic matter, the zooplankton, and moving up to the rest of the food web. So if this is the one you choose to draw, that'd be okay, but I have a different one as well. So you can choose between this one and the next one I'll show you. I'll tell you when it comes. All right, so we just see the same thing right here, and the bacterioplankton, right? So they're bacteria, and they're floating as well. Our
The microbes in the ocean are the very small organisms that we can't see with the naked eye. We start with the smallest, they're the viruses, very, very tiny. And if we take a teaspoon of water as a measure, then we're looking at many millions of viruses in a teaspoon of water from the ocean. And if we go the next step up, we start to think about the bacteria. Again, very small, microscopic, around about 2 to 5 million in a teaspoon of water. If we go to the next size up, we're starting to look at the algae or the small single-cell plants that we call phytoplankton. And in a teaspoon of water, we're looking at several thousand to maybe 30, 40, 50,000. And then if we go the next step up of the microbes, we get into the very small microscopic animals that eat the bacteria and the small phytoplankton. And there we're looking at, in a teaspoon of water, 1 to 2 in a teaspoon of water. The microbial loop in the ocean is all the microbial organisms and their relationships to each other in terms of their interactions in the food web. The microbial loop forms the very bottom of the food web with all the microscopic organisms. So we have the bacteria taking up organic material from the water and growing. We have the algae using sunlight to photosynthesize and grow, and then we have the microzooplankton that then eat the bacteria and the algae. And those microzooplankton then release in their feces nutrients like nitrogen and phosphorus that are really important for the growth of the bacteria and the algae, and they recycle that back into the system. We've got to remember also that once we get out of that microbial loop, then those small microzooplankton are eaten by larger zooplankton, and that then goes up through the food web to the larger organisms, to fish and marine mammals in the Southern Ocean. Also, you get the cycling of carbon in the water. So you get release of organic carbon by the algae and also the microzooplankton, and that's taken up by the bacteria. So you get quite a quick cycling of both nutrients and carbon within the microbial loop. And that's really important in terms of how fast things are moving and cycling within the ocean. And also when you have a dominance of the microbial loop in the ocean, you tend to get a lot of recycling in the upper waters, and then you don't get as much carbon, the big algae and the larger zooplankton growing with lots of carbon sinking out into the deep ocean, what we call carbon sequestration, where you take carbon out of the atmosphere into the water, it's used by the algae and then taken into the deep ocean.
All right, so this is the one I was thinking you could draw. This would be the easier one to draw, maybe. I like the way on the right there it shows sort of that loop where our bacteria, are kind of making that loop all the way around. They're sort of doing their own thing, but the rest of the life is benefiting from it. So that's what I was kind of thinking. You can use this one. You can use the previous one if you like. There's even one in your book, I guess, if you want to try to draw that one. So you can pause this right here and draw that out at your own pace. All right, so the food chain, here's what we think about. Look at the bottom there, all that phytoplankton down there, the primary producers, and then the zooplankton, then we have fish and bigger fish, and of course the apex predator there at the top. So remember, there's no place to hide, so they have to use their other strategies. Some sort of camouflage like clear stuff. The speed, of course, is big. Counter shading is big. And then we didn't talk about this, but as far as a type of camo would be using false eyes, stripes, things that will confuse the predator. So you've seen like the redfish, for instance, has a big spot at the end of its tail that looks like an eye. And so a predator might think that's its head when really it's its tail, and it gets the redfish can get away that way. All right, now we're on primary productivity and nutrients. I believe you turn the page now. You're on 89, page 89, and you're going to write these few things down because we're talking about primary productivity. Light, of course, is the major factor of the production rate here. We need to make sure there's plenty of sunlight for our organisms to photosynthesize. We need nitrogen and phosphorus to help with that process. Even though it's not used in the chemical reaction, it is indeed makes the creature stronger. And the water must be moving. Stagnant water is never good. It tends to use up the oxygen and not add it back in. And so if we keep it moving, we can get that oxygen adding back in, and also carbon dioxide from the atmosphere added back in, so that the photosynthesis can take place. This is a map showing the primary production around the world. The red parts right there you can see is where it's the strongest. It's very full right here all along the coast. And you can guess why that's true, and it's because of the warmth, right? And the shallowness of the water, that these creatures these phytoplankton can be very, very close to the sun, and they can produce a lot. Notice the rest of the ocean has some activity in it because those floaters in there are also going through photosynthesis. But we also notice that there's going to be movement among these areas right here. So the major thing we're looking at here is called overturn, how the water's going to turn itself. We're going to see how the temperature of the water, the winds, and the Coriolis effect all will help with mixing the waters up and to keep them going so that we have plenty of gases being dissolved in there. So the two systems that we're going to be talking about here, the processes that allow nutrients to move throughout the water column, those would be overturn and upwelling. So overturn is when cold water, which sinks, it pushes up warmer water, which is rich in nutrients, to the water column at the polar regions. So in the polar regions where the cold water is, it's going to sort of make a loop down there as it's pushing down and up the warmer water. And the warmer water is not that much warmer, but it's warmer than the ice cold water. And then upwelling sort of toward the equator, you will see this. You will see the winds that have a lot more activity in this, more than the temperature. But colder water will actually be kind of sort of, if you want to think of it as being pulled up to the shallower areas by that wind blowing, by that Coriolis effect that's happening there, causing the upwelling. This picture kind of shows the upwelling there. See, we're at the equator, and the cool water will actually sort of be pulled up by the action of the above surface. The warm water surface will be blowing away, and as it blows, it causes the cooler air to move up and take its place, and causing motion, causing movement in the water, making it nice and healthy. You've probably heard of El Nino, so it's a phenomenon that changes the upwelling patterns. So normally you can see in this top picture right here, we have just the heating of the water that comes off the coast. So we have cold water around here moving upward, and then it's going to move over toward Australia this way like this. And then we have more heating of the water, the warm water, which is moving the other way during an El Nino year. Okay, it'll come up here. It actually affects North America quite a bit. We have a warmer winter and a much wetter winter typically. So it's related to this thing called the Southern Oscillation, and that has to do also with the Coriolis that's moving along there. So it describes a pressure gradient between the Pacific and the Indian Oceans, and we can make predictions based on the temperature and obviously the flow if we're going to have an El Nino year. I'm not sure if we know when they're coming, or we can just say by looking at the data, oh my goodness, this year is going to be an El Nino. And the reason why it matters is because it will cause a lot more rain for us. And so it's good for us to know that it'll be warmer, wetter winter if we're having an El Nino year. So we're going to write this down right here. It's a change in the surface current on the coast of Peru and Chile. The Western Pacific water is warmer than normal. The upwelling decreases near South America, creates more rain for the northern coast of North America. Texas in particular, we get a lot of rain over the, even over the desert area as well. Interesting note here is, why is it called El Nino? It turns out that the Spanish-speaking people in the Southern Hemisphere noticed this and made the correlation of this warm water around Christmas time. And so El Nino actually refers to the boy, the little boy, the baby boy, namely baby Jesus. So that's why it's called El Nino. So there is an opposite effect, similar but opposite, is when we have cooler waters coming off, and we call that La Nina. It's just a complement to El Nino, little boy, La Nina, little girl. And so we see what happens here is the water is cooler, and it creates drier conditions, drier and cooler conditions in North America. So we can have droughts. Once again, they only predict this after they test the temperature of the waters. This is a great picture showing an El Nino. You can see this right here is very warm water, and that warm water is coming off this coast right here, moving this way, right? And that will cause the heating of all this water. Coriolis is going to swing all that moisture this way in the air and then come right down over Texas and give us a nice, wet, warm winter. Okay, so before we end this module, I want to spend a little bit of time talking about whales. We haven't spent a whole lot of time talking about them. And so since we mentioned them earlier, talking about large nekton in the ocean, I want to spend a little time on here. Here's the humpback whale. It's a pretty fascinating creature. They are ones that migrate pretty regularly along our western coast there, and they're really fun to watch. When they dive below the surface, they lift their tail fluke out of the water. So they show the special tail fluke. So you can see this guy going under and the tail coming up, and on the right side there, that part of the tail. Well, what's interesting about this fluke is that each whale has an individual marking on their fluke. It's sort of like a fingerprint. And so those people who are out there studying whales, doing the marine biology studies out in the ocean, they actually name these guys, and they can look at the tail, probably take a picture of it because it happens so quickly, and they can tell what whale it is, and therefore they can keep track of them and see how their migration is doing and how well they're doing. So if we're looking at flukes, you can see here all the things. They range. Some of them are all white. Some of them are all black. Everything in between. And you can see how there's various little changes that take place. And so they call them things like eyes and the trailing edge and fireworks and stuff like that. I don't know if I've seen one of those right here. You can see with the fireworks on them. That might be kind of fun. And so depending on what critter it is, they have a unique design there, and they get named that way. We have white on the edge. We have straight-sided right here. This part is straight-sided, right? Rounded, not all the way to the notch right here, like that. And then these groups are sort of variations of the ones above them, like that. So it's kind of like a birthmark. And like I said, the marine biologist can say, oh, there goes, whatever the name of that whale is. Pretty cool. So here's one. So you're going to pick 2 or 3 special markings on the fluke picture. Okay, so you can kind of see it's not completely black, right? We have the white on the edges there. Okay, and we're going to look at that, kind of that notch, and that one kind of coming up and a little bump. Oh, look, I don't know, it looks like a scratch or something, doesn't it? Okay, so remember that in your mind right there. We're going to match it. So let's see who it is. All right. I'm checking it out. What do y'all think? Who do you think that was? You can go back. I don't think I can, but you can go back and take a look and see what you think it matches to. I think I get the answer here, though, in a minute. Yeah, I was kind of guessing springboard. Yeah, right. So you make the match, then you can learn the name of your humpback whale. Okay, so this whale's name was springboard. Very cool. Y'all can go back and look at that and see if they do have those similarities there. Okay, it's lab time. So this is a lab that it's about the floating situation. So you're going to kind of have that one in the book with the clay if you want to do that. This one is more like using the air bladder type of a thing and the pressure. So let's watch this video and see. All right. Hi there. Good morning. All right. So what I have here is the experiment we're doing for this lab. It is a short experiment, and it's showing, we're talking about these creatures and how they are able to navigate up and down in the water column based on their buoyancy. And sometimes they want to sink, and when they do the vertical migration, of course, at nighttime, they will rise to the top and come up and eat, and then go back down to the bottom in the daytime. It's for protection, really. And so we're going to show how one of the strategies for that. We've seen several, like the air bladder in a fish, and the oil droplet in some of those little creatures, and the sail, and many different ways of keeping these creatures buoyant. If they get too deep, they'll get away from the food source and all that kind of stuff. So what I have here is this little creature. You can see him a little bit there. It's called a jellyfish or a squid or whatever. And it has a little hole in the bottom right there. So it's just like a little, I have to work out this camera thing. It's just like a little chamber in there. And so he's made of plastic. You can see through him. And I'm going to drop him into this bottle of water. And he's going to float because he's plastic. There he goes, turns right side up. There we go. And I'm going to put the lid on right here. And so now this creature is floating up in the upper part of the water column right here. And a strategy to get him to come back down would be to actually increase the water inside of him. So this is called a Cartesian diver, by the way, named after Rene Descartes, who supposedly came up with the concept or something like this. And so what we're going to do, it's a matter of pressures. So we're going to build up pressure on the outside of this critter, which will push the water up into the critter, and therefore we're going to see what happens. So any predictions what's going to happen? I'm sure you're predicting it already. What's going to happen when I squeeze more water into the creature by putting pressure? Now, if the lid were off, this would not work because the pressure wouldn't be there. This guy would pop right out of the bottle. But we're going to see what happens when I squeeze it. Okay, so I have to kind of squeeze it hard. You can see his little face there. Let's see what happens. There he goes. All right. See, he's coming down. He'll actually go all the way to the bottom there. And then when I release, guess what? There we go. So think of it like as an air bubble inside of him. And I'm making that air bubble smaller when I introduce more water. So it's kind of fun to do this. You can push down a little bit and then maybe stop midway through, make him do a little dance and go up like that. We can go all the way down and then come up kind of slowly just by manipulating the amount of pressure that's on there. There we go. All right, so that's what y'all would have been playing with today. We could make another one with a test tube, and we would put the test tube inverted inside the bottle. I have one of those big 3-liter bottles. And when we had that inverted in there, with a little bit of water in the test tube, you can actually see the water going up in the test tube. The problem is that big bottle right there, I'm not strong enough to push it. Some of y'all would have been, would have got y'all in there really pushing hard and that kind of thing to see that amount of pressure. So much pressure on the outside of that bottle when all that water is in there, because water is very, very heavy and strong. All right, so this is our lab today. I think that's what you're supposed to write up in your book. And we'll see you again next time. So long, y'all. So remember to finish Module 13, do your study guide. I'll have to figure out a way to give you credit for that. I'll just get your parents to okay it. I think there's a thing on this Google Classroom that we're going to use that allows you to input. You can even scan things in there. And I think there's a quiz. We're supposed to have a quiz next week. So I think there's even a way to take a quiz online. So we'll have to see about that. I'm still experimenting with Google Classroom, but we will get that worked out. Okay, everybody. Thanks so much. I hope it worked. Let me know. See ya.